our next speaker is Tian. Yeah. Taylor is going to show us a package demo on Stereopy as an advanced tool for interpreting spatial transcriptomics data. So let's thank Taylor. Let's welcome Taylor.
Hello. Hi, my name is Taylor. Sorry, a little bit cough. It's great to have this opportunity to talk a little bit about the Stereomics solution of BGI research. So the name is Stereo-seq. It's a large field of spatial transcriptome solutions. We got the CID, we call it CID, which is the position information, and pre-sequenced. When you receive the chip, the information is also come with this chip and can be used for the analysis of your data to locate the sequence back to the position. And after the CID, we have UMI and polity to capture the transcriptome. And then we use the in situ RNA capture to capture the whole region of the organ, and then to make it to the DMB, like put it and make it into the DMB and do the sequencing. And after that, we use the downstream analysis to do the cluster and sort of analysis to let every rays go back to the original position. So two things are very important for the spatial solution. One is the resolution. As I showed before, the resolution is very small. And another one is the capture region. It can, our solution comes from 50 millimeters square to the very big size, which can cover the whole monkey semi-brain section. And we have some papers coming out. The upper left one is our methodology one, and I will show a little bit more information about this part. The embryo development is very important for the mouse study or for the development study. So we select some of these organs for the sequencing and analysis. Here is a result. You can find this result in the first page of the paper. The time is from E9.5 to the E16.5 to get better idea about the very big mouth. We also has 13 slides for the E16.5 to have better idea about the different parts. And from the gene expression result, we can see that different organs like the brain muscles, they have a very distinguished hotspot expression from the figure. And also for the gene regulatory network, we can see some genes has very differentially expressed in different part of the body. And to make the analysis more comprehensive, we can also do the vertical analysis of some genes for the mouse heart, and also the artery heart of the mouse to have better idea about the hotspots genes expression. So with the spatial information, actually this is like the analysis based on the image and also the sequencing. With this, we can have a very optimized clustering result. And if you go to the right video, you can see that we can go to very detailed position and information about the mouse organs, different part. And also to test the reproducibility, we sequenced two mouth olfactory bulbs, the section 1 and section 2. And we compare the UI mice and genes, and we found out the reproducibility. Like here, we can see that the reproducibility is very high. It's about the 96. All of the above things were analysis by two of our in-house pipelines. The first one is SAW, which is software. You can find that in the GitHub, and also the Stereopy. You can also find that in the GitHub. The left one was for some basic analysis, and the right one is more for the visualization. It's more like a toolkit. You can have a lot of different functions like cell segmentation and visualization and clustering selection, sort of like the right one. You can do a lot of different small functions to have better idea about your result. And if you are of any interest, you can go to this part to have a demo data downloaded and to do the analysis. And normally, if you want to do the spatial sequencing, you need to do a very, very big sequencing and analysis. And like MGI machine, we have the very powerful machine and sequencing machine and analysis server, which enables the sequencing and analysis of 50 human genomes within 1 day. So very welcome everyone to collaborate with us to find a better solution of the spatial analysis in the server to make it better adapt to the spatial analysis. And if you have any more questions, welcome of any questions, but if you have some more detailed questions, you can go to this email for more details. Thank you.
Thank you, Taylor. Are there any questions for the speaker? There are any questions from, I have one quick question. Mine is a more general one. So I know that we have been hearing before a lot about bioconductor packages. So in bioconductor, we often have data structure like single cell experiments and for spatial data, there are other ones as well. Is there something similar that you would use in a Python package?
Yeah. I think, but actually, for the single cell, I think most of them are ours, and we haven't done too much Python work on single cell, because we developed this one just because we need a very big power or very memory assumable to do the analysis. So we developed the Python one, but I think there should be some.
Okay, thank you. Any other question? Otherwise, let's thank again Taylor.
Thank you.